Thank you for joining us today for our first webinar in our new Creating a Better Future series. Today, we'll be discussing the future outlook of food allergy therapies. I'm Jennifer Gertz, Executive Director of Food Allergy Canada, and our guest speaker today is Dr. Cecilia Berin. First, just a bit about Food Allergy Canada. As many of you are aware, we're a national nonprofit charity and Canada's leading patient organization committed to educating, supporting, and advocating for the more than 2 million Canadians living with food allergy. 100% of our funding to support these efforts comes from the generosity of individual donors, corporate sponsors, and a few foundations. We'd invite you to consider Food Allergy Canada in your charitable giving to support sustainable educational sessions like today's session. So our webinar today is about food allergy therapies that are in the development cycle that are happening now, and also we'll cover what's in the pipeline for future possibilities. This was going to be one of our sessions at our national conference in May, but unfortunately we had to cancel that due to COVID-19. However, we're excited that we're able to transform the presentation into an online webinar so all of you can benefit from the insights that Dr. Berin is going to share. I'd also like to thank everyone who submitted questions in advance on registration. There were a lot of really great questions, and we might not be able to get to all of them during this presentation. However, know that we keep those questions. It provides us with a really rich view of what's on everybody's minds and helps us inform webinar content that we should consider going forward. A couple of housekeeping notes before we get started. Please note that Dr. Berin's presentation is for informational purposes only and will not provide specific medical advice, recommendations, diagnosis, and treatment. All participants are muted so we can keep the audio clear for the webinar. If you have any questions during the presentation, please submit them in the chat question box throughout the webinar, and we'll try to get to as many questions as we can. And lastly, the webinar will be recorded and it will be shared on foodallergycanada.ca afterwards in case you want to refer back to it or you'd like to share it with others. Now I'd like to introduce Dr. Cecilia Berin. She is a Professor of Pediatrics and the Deputy Director of the Jaffe Food Allergy Institute in New York City. Dr. Berin's research focuses on understanding immune mechanisms of allergy and tolerance to foods. Her lab works with clinical trials to understand why some individuals respond better than others to therapy, with the goal of developing better treatment approaches for food allergy. Her lab group is also working on the immune system in early life to understand susceptibility to food allergy in infancy. Dr. Berin's work is funded by the National Institute of Health. Dr. Berin, I'd like to turn it over to you to walk us through the future outlook of therapies.
Thank you very much for that introduction, Jennifer. It's a pleasure to be here. So I'm speaking to you today from a town just north of New York City. My thoughts are with everyone who is struggling right now in so many ways from this pandemic. I was very happy to be asked to prepare this webinar and turn my thoughts to food allergy and to share with you some of the major strides that have been made in this field. Okay, so I have a couple of disclosures. I serve on the advisory board for Therapeutics and have received payment for speaking at a DBV Technologies sponsored symposium. I have made every effort to make this presentation today balanced. Today, I will be discussing different approaches to the development of new food allergy therapies, including immunotherapies present and future, the use of a new category of biologic drugs to modify the immune response, and finally, the use of microbial therapeutics to treat food allergy. I'd like to start by briefly discussing how the immune system causes food allergic reactions. And at the center of this disease is an antibody, a type of antibody called IgE. So IgE is normally made to protect us from parasites, but when it is recognizing innocuous things like the food that we eat, it can cause serious symptoms. IgE antibodies sit on the surface of cells. They coat these cells called mast cells, and these cells sit throughout tissues all over our body and are particularly enriched in sites that have contact with our environment, like our skin, our lungs, and our gut. When a food allergen comes into contact with IgE that recognizes it, and it binds to 2 of those antibodies to bring them close together. That process activates the mast cells, and those mast cells release many different kinds of mediators. Those mediators can then act on cells throughout our body, blood vessels that can vasodilate, smooth muscle, epithelial cells that line our lung and our intestine. This can cause a diverse set of symptoms that are triggered by those foods. So, such as hives in the skin, coughing and trouble breathing in the lungs, or nausea, diarrhea, or abdominal pain. So the goal of therapy is to prevent this type of immune response from happening. Let's talk a little bit about the process of research that leads to new therapies. So generally, new ideas start in the lab. And as we understand more about the cells and the pathways that lead to disease, we get a better idea of how we may be able to target those pathways to then prevent or treat that disease. So these preclinical studies often use mouse models of disease, mouse models of food allergy, or they use cells derived from patients to test if that drug or treatment approach might be effective. And then there is this extensive phase of human studies. And this human testing is done to make sure that drugs are both safe and effective. Phase 1 studies are done first. These are really aimed at safety. Phase 2 studies are expanded safety studies that can assess also how well the drug works. And phase 3 studies are large-scale studies that are done to test whether or not a drug works in a large, more generalizable population. So this is really critical. This phase 3 positive results in a phase 3 trial can be used to generate an application for regulatory approval. And if granted, then there is further follow-up safety data gathered. So in general, what kind of time does it take to go from start to approval? And I'm going to give you 3 examples of the research process specifically in food allergy. I'm going to start with one example, and this is a peanut vaccine. This was developed by my colleague, Dr. Hugh Sampson at Mount Sinai. And this drug development process was done entirely within the confines of academia at the university. And this started out in the early 2000s. There were several key findings that really came together. First of all, they developed a mouse model of peanut allergy, very critical for testing some of these new therapeutics. Then they did a number of studies to identify where on the peanut proteins the IgE antibodies bound. And they developed these modified peanut proteins that were hypoallergenic variants. Then they did a lot of work to package these proteins. They developed E. coli bacteria that produced these modified peanut proteins. And then they did a proof of concept study in mice. So these peanut allergic mice, they treated them with this vaccine by intrarectal delivery. It worked really nicely at preventing anaphylaxis. And then they went into the study planning process for human trials. And in 2009 and 10, a phase 1 study was done. This was a multi-center study that was run through COFAR, which is a consortium of food allergy research based in the US. And results were reported in 2013. So unfortunately, this timeline of therapy development stopped here after the phase 1 study. And it was because during this phase 1 safety study, it was found that there were significant reactions to the therapy itself, which really prevented, even though these peanut proteins had been modified to be hypoallergenic, this really did not look so promising for further development. So without a drug company behind it, this process took about 13 years from initiation to termination. The next example I'd like to give you is about epicutaneous immunotherapy. So this is also known as EPIT, or the peanut patch. And this development was driven entirely by the drug company that developed it. So this company developed this ViaSkin patch, and first published a number of preclinical studies. These were all done in mouse models of peanut allergy from 2011 to 2015. And by 2016, they had gathered results and published their phase 1 study. One more year, and two phase 2 studies were published. And by 2019, the phase 3 study was completed, analyzed, published, and they have submitted an application for regulatory, for FDA approval. And this is pending. So in this example, approximately a decade from conception to approval, if that approval is in fact granted. And the third example I'd like to give you is a therapy that was recently approved. And this is for peanut oral immunotherapy. And here, unlike the first two, there really wasn't a preclinical phase. Oral immunotherapy had been reported in the literature going back to the early 1900s. And in 2009, Wesley Burks and Stacy Jones published two studies that showed that peanut oral immunotherapy was both safe and effective in these small single center studies. And they followed that up with a placebo-controlled randomized study of peanut oral immunotherapy. So in 2011, the company Aimmune was founded. And this was formed for the sole purpose of getting peanut oral immunotherapy available as an approved treatment for food allergy. The Food Allergy Initiative, which is now FAIR, a patient advocacy group, was the major driver in making this happen. So they were really focused on getting the clinical trials done. Phase 2 and 3 trials followed. And with positive results in this phase 3 trial in 2019, they applied for, actually that was 2018, that phase 3 trial was published. They applied for and received FDA approval at the start of this year for a peanut OIT product that they call Palforzia. So again, approximately 10 minutes from start, and here the start within academia and finished by biopharma. And at the end of this webinar, I think we'll have some time to talk about what the Palforzia approval means for care in Canada. So now I'd like to talk in a little more detail about these immunotherapies, starting with Palforzia, which is what I just talked about. This is oral immunotherapy treatment for peanut allergy, again, just approved this year in the US. So what is oral immunotherapy? It is a desensitization procedure in which starting with very small amounts of food protein, that this is built up over a period of many weeks, slowly and incrementally until a maintenance dose is reached. And this therapy protected most individuals that were treated from a dose of up to 2 peanuts. So here, 600 milligrams is a dose of 2 peanuts. This is the percentage of those who were treated who could tolerate that dose. You can see that more than 50% can tolerate this higher dose of peanut versus the individuals with peanut allergy who were treated with placebo. Really a very small minority of them could tolerate those higher doses of peanut. So this treatment is meant to be continued for as long as that desensitization or protection from higher doses of peanut is desired. So Palforzia is a specific peanut oral immunotherapy product, but there have been many studies on oral immunotherapy for other foods, including milk, egg, wheat, and others. So in summarizing basically all of the publications that have come out on oral immunotherapy, generally 75 to 80% of individuals who were treated with the oral immunotherapy can reach a maintenance dose and become desensitized to that food. So what are some of these terms? What do I mean when I talk about desensitized? I mean able to tolerate exposure to the food while taking the daily therapy. There is a lot of interest in whether oral immunotherapy can lead to lasting changes. The term tolerance is a term it's used in immunology. It's quite hard to demonstrate. It's a technical term, basically meaning that the immune response is actively preventing a reaction to food. But again, it can be difficult to demonstrate that in individuals because it's basically a permanent state. Instead, the term sustained unresponsiveness has been used a lot in clinical trials. And this is a term that's used to indicate that individuals can avoid the food for a period of time, and generally clinical trials test this for about 2 months. And at the end of that 2 months of avoiding therapy, they're still able to tolerate the food. So in general, lower frequency of individuals can develop sustained unresponsiveness compared to being desensitized. But for some foods, for example, with egg, there has been relatively good long-term success after oral immunotherapy, with most individuals who reach that desensitized and sustained unresponsiveness stage able to incorporate egg freely into the diet. But there's still a lot of questions about OIT in the long term, what this means for long-term management. In general, it's thought that there needs to be long-term regular ingestion of the food in order to maintain desensitization. So as with any therapy, there are pros and cons to the treatment. We know, as I mentioned, that OIT does successfully desensitize most patients who are treated. And some of those over time will eventually be able to incorporate that food into the diet. Now, the cons are, first of all, for most patients treated, there are reactions to the therapy itself. And in general, these are mild. These mostly occur during the buildup phase. But for some, these are more severe. For approximately 15% of patients tested in clinical trials, they have to drop out of the trials because they have severe persistent gastrointestinal symptoms. It's been documented that approximately 2% of patients treated with oral immunotherapy develop inflammation of the esophagus that's called eosinophilic esophagitis. The symptoms resolve on termination of therapy, but again, this is a concern. One other con for those who have multiple food allergies is that this therapy only desensitizes to the food given. For example, an individual who is receiving peanut oral immunotherapy will not be protected from their allergies to tree nuts. One of the major limitations that I mentioned of oral immunotherapy is the fact that it does cause reactions. The same immune reactions that cause food allergic reactions can be activated by this oral immunotherapy. So one approach has been to use alternative routes of immunotherapy. And these appear to induce fewer or less severe reactions. Sublingual immunotherapy, which is commonly referred to as SLIT, involves giving small amounts of food antigen under the tongue. And this is generally 100 or 1,000-fold less of the allergen than is given as oral immunotherapy. Epicutaneous immunotherapy, or EPIT, involves giving even less allergen, so roughly 1/4 of a milligram of the allergen topically on the skin, and this is delivered by a unique patch called ViaSkin. So both of these routes involve much less allergen than oral immunotherapy, and while reactions can be triggered, they are generally local, such as mouth itch for sublingual or itchy skin under the patch. Now, in terms of effect, this is an important trade-off, and oral immunotherapy, as shown here, generally provides the greatest level of desensitization, but that comes with the greatest frequency and severity of side effects, while SLIT is somewhere in between, and EPIT has the lowest level of threshold reached, and most lowest frequency of side effects. So again, this is a trade-off that needs to be considered when considering any of these routes of allergen immunotherapy. Now, there are other variations on immunotherapy that use modified forms of allergen that trigger fewer reactions. One alternative to oral immunotherapy is the use of baked foods, such as baked egg or baked milk. We know that proteins in milk or egg are changed by high heat, and when they're unfolded in this way, they're less able to bind to IgE antibodies. And because of this, baked versions of the foods are tolerated by many with milk or egg allergy. And the inclusion of these into the diet appears to have a modest effect on promoting outgrowth of milk or egg allergy. There's also a company called How Allergy that has developed a product that involves chemically modified peanut allergens. So these are not heat treated. Peanut allergens are generally resistant to heat treatment, but they are chemically modified to again unfold them and make them less able to bind IgE antibodies. These are provided as an injection, so similar to allergy shots for grass or tree pollen. And this approach is being tested in clinical trials right now. And finally, an approach called peptide immunotherapy has been proposed. And instead of giving the whole protein, there's interest in using short fragments of the food allergen. These short fragments or peptides can't physically bind to IgE antibodies at once. And because of that, they're not able to cross-link IgE and they can't activate the mast cells. So these approaches all share the same concept of providing the antigen in a slow and safe way with modified allergens that are less able to activate those mast cells through IgE. So what do we know about the mechanism of how immunotherapies work? So the slow, steady exposure to allergen as immunotherapy generates a different kind of antibody. And these antibodies are called IgG and IgA antibodies, and these are protective. These are the kinds of antibodies that protect us from things like bacterial or viral infections. And how they work in food allergy is that they compete with IgE antibodies for binding to the allergen. And some of these antibodies can also act directly on the mast cells to inhibit them from getting activated. So what immunotherapy does is it adds a protective immune response on top of our allergic immune response. You know, in some cases, there's also a reduction of the amount of IgE that is present. Another approach to treating food allergy is to, in fact, directly target the IgE that causes reactions in the first place. And this is done with a drug that is in the category called biologic. This is called omalizumab or Xolair. And biologics are drugs that are engineered parts of the immune response. So just like we can make protective antibodies against the flu, in the lab, we can engineer protective antibodies. And omalizumab is an antibody that is engineered to bind to IgE antibodies and remove them from the body. So omalizumab has been proposed to be used in food allergy in two different ways. And the first is as a standalone therapy or monotherapy. It removes the IgE from the circulation. It eventually removes the IgE from the surface of those mast cells. And now there is basically no ability to activate those mast cells through IgE. Now, because this approach is not food specific, this should work for all allergens, theoretically. Because it doesn't stop IgE from being made, this has to be given, either given indefinitely, or food has to be introduced into the diet under protection of omalizumab acting as a desensitizing or oral immunotherapy with the possibility of omalizumab then being tapered off. So the other major way that omalizumab is proposed for use in food allergy is together with oral immunotherapy. So in this approach, omalizumab would be started, then oral immunotherapy to either a single or multiple foods could be started. And the idea here is that omalizumab would prevent the reactions that are commonly induced with oral immunotherapy. And then eventually there would be weaning off omalizumab as oral immunotherapy had been established. So these two approaches are currently being tested in a large multi-center clinical trial in the US. Overall, the pros of omalizumab is the potential for action on multi-food allergens. The con is that it doesn't stop IgE from being made, so it may have to be continued indefinitely, and certainly the transition from omalizumab to food in the diet or continual oral immunotherapy needs to be studied. So omalizumab binds to IgE, reduces the level, but it doesn't prevent IgE from being made, which is why it has to be maintained as a treatment. So next on the horizon are treatments that could interfere with IgE being maintained. So what do we know about the immunology of IgE? We know that cells called B cells that are shown here are the cells that make antibodies. They make IgE antibodies, but they also make protective antibodies like IgG and IgA. So how do they decide what kind of antibody to make? They get instruction from cells called T helper cells. These are shown here. And these T helper cells produce cytokines. This is a type of protein that immune cells use to communicate with each other. And the cytokine that tells B cells to make IgE is a cytokine called IL-4. So there is now a drug on the market for other allergic diseases called dupilumab. This is also a biologic like omalizumab, and this blocks the action of IL-4 and the related cytokine IL-13. Okay, so this, again, it's an antibody. It binds to the IL-4 receptor, one chain of the IL-4 receptor, which is also part of the IL-13 receptor. It prevents IL-4 or IL-13 from being able to bind to this receptor. So theoretically, this antibody should prevent IL-4 from instructing B cells to make IgE. So what is the evidence that this might work for food allergy? Well, I mentioned that this has been approved for other allergic diseases, so other clinical trials have been done. And what I'm showing here is data from a clinical trial with asthma and another one that was done with a nasal allergic inflammation. And shown here are placebo groups on top versus dupilumab, and this is our charts that are showing the amount of IgE in the blood. And you can see that over time with treatment with dupilumab, there is a reduction in the level of IgE in the circulation. So if it interferes with IgE being made, this could potentially reprogram the immune response to prevent food allergy. There are currently at least 3 clinical trials that are ongoing with this drug, dupilumab. So one of them is using dupilumab alone. Again, this is called a monotherapy. And this is being done at several sites throughout the US and in Canada. A second approach is using dupilumab together with oral immunotherapy. And this is using that Palforzia immunotherapy, peanut immunotherapy. And this is being done in a number of clinical trials throughout the US only. And then there is a third trial that is being done at Stanford University, and it is comparing the combined effect of dupilumab and omalizumab. So I think in the next few years, we're going to have a lot of information about the potential use of this, I think, exciting new drug for treatment of food allergy. So as we have learned more and more about the immunology of food allergy and how IgE gets generated, there are more and more drugs in the pipeline. And shown here is a schematic of this pathway, this immune pathway that leads to the production of IgE. I mentioned that T cells instruct B cells to make IgE. T cells receive their instructions from dendritic cells, and they receive their instructions from these epithelial cells through many different mediators. What's exciting now is that there are a lot of different biologics, dupilumab I mentioned. There are also a number of small molecule inhibitors, so these are called JAK inhibitors that inhibit the signaling downstream of this IL-4 and IL-13 receptor. There are antibodies that target this interaction between dendritic cells and T cells. There are antibodies that actually target the B cells themselves. So there are a lot of new biologics, a lot of new small molecule drugs that can be given orally. Many of these are in the pipeline for other allergic diseases. So there's already some safety data on these drugs. So I think, what's exciting is that there are many other drugs that are lined up behind dupilumab for, I think, quite rapid testing in food allergy. So now I'd like to switch gears to a completely different class of therapy. And this deals with using our gut microbiota to treat food allergy. So it's been known for quite a long time that there are certain lifestyle factors that influence our risk of developing food allergy in early life. So it's been known, a number of studies have shown that having a dog in the house in the first year of life is protective for food allergy. Children who grow up on farms early in life, where those farms have livestock, are protected against not just food allergy, but many allergic diseases. There are also factors like mode of delivery. So vaginal birth is protective against development of food allergy. Having more older siblings, attending communal childcare. These are all things that have been documented as changing the risk of developing food allergy. And what all of these things have in common is that through their environmental exposures, these change our microbiome. So what is the microbiome? The microbiome is the community of microorganisms that inhabit our body. So primarily we think about the microbiome of our gut, and this is the area where most of the organisms of our body live, but there are also healthy communities of microorganisms in our lungs and on our skin. They're basically all over our body. And we know that the human body contains 10 times as many bacterial cells as human cells. So there's a huge amount of interaction between the microbial world and our cells. And we know from many studies in mice, and these studies have used mice that completely lack bacteria. So they're grown in isolators. They are referred to as germ-free mice. And those studies have shown that our normal bacteria have an immense influence on our development for all sorts of systems, but is really essential for the development of a normal immune system. So how does the microbiome contribute to food allergy? Well, we know from studies in young infants and children that the microbiome is different in those with and without food allergy. So these studies have been done by taking stool samples and by high throughput sequencing of the genes. So initially studies were done by culturing bacteria. Now we can do this really high throughput way of by looking at the bacterial DNA, identifying all of the different bacteria that are present in the stool. And using this approach, again, it was shown that food allergic and non-allergic kids had different microbiota. And we know that importantly, this difference precedes the development of food allergy. So this was first identified in the CHILD study, which is a large study based in Canada that is looking at predisposing factors to allergy development. So why is that important? it shows that it's not just a matter of difference in the diet. So for example, if kids are avoiding milk, it can profoundly change their microbiota. But this showed that the composition of that microbiota was different before these children started showing any symptoms. So is supportive of the idea that this may be causative. Another important study showed that milk allergic children who outgrow their milk allergy have a different bacterial composition than those who have persistent milk allergy. So why do I think this study is important? It points to a role for bacteria not only in getting allergy, but in resolving it, which I think is very important if we're going to think about using bacteria as a treatment. So now I'd like to go back and tell you a little more about the studies that have really shown the importance of the gut microbiota in food allergy. And this work started using studies of mouse strains that were either resistant or susceptible to food allergy. So these were genetically distinct strains that had this different susceptibility to food allergy. And scientists found that when looking at the composition of the microbiota in their stools, that they had a different set of microorganisms in their gut. So they then transferred this bacteria to new mice through a process called FMT, or fecal microbial transfer. And mice that received bacteria from susceptible mice became susceptible, while mice that received bacteria from resistant mice became resistant. So these were genetically identical germ-free mice to start with, and only the transfer of that microbiome from these different strains of mice led to those different outcomes here. So similar studies have been performed, and here, instead of using susceptible and resistant strains of mice as donors for this FMT, here the donors were food allergic and non-allergic infants. So we already know that their gut microbiota composition differs. And those studies that show that the microbiome differs, they have no way of telling whether that difference is functionally meaningful. But here you can actually test this by, again, putting these into germ-free mice. And what they found when they did this was that this transfer into germ-free mice led these mice to become either resistant or susceptible, based on the clinical group that the bacteria came from to begin with. And finally, I think some really exciting data comes from these experiments in which they added back the bacteria that were missing from food allergic individuals. So they found that there were differences in these strains, the Clostridiales or Bacteroideales species. And they then tested if they add these back, will it make a difference to the outcome in the transfer of these mice? And that's what they did. And shown here is the outcome, that adding back these strains to the stool from food allergic infants then rendered these mice resistant to the development of food allergy. So this is really the basis for the idea that giving certain microbes may be beneficial in treating or preventing food allergy. And there's been really an explosion of companies that have been created to go after this very idea. Okay, so there are currently many different approaches to modifying the microbiome in clinical trials. So starting out with the diet, we certainly know that the diet is one of the most important factors in determining the composition of the microbiota. And there are certain components of the diet, like dietary fiber, that have been shown to be protective for a number of intestinal diseases, including food allergy. So probiotics are another major category. These are being proposed for food allergy treatment, and these are a little different than the standard probiotics that are widely available. And these are bacteria that have been identified as being missing or lower in the context of food allergy. And so I see these as a second generation type of probiotic, really identified by showing what's missing in the context of food allergy. There's also a category called prebiotics. These are compounds such as oligosaccharides that are utilized by certain bacteria and can promote their growth. While the category of symbiotics is a combination of probiotic and prebiotic. And in the most crude form, fecal microbial transfer, or FMT, is being tested in humans. And there's currently a trial going on in the context of food allergy for adults, looking at what is the impact of FMT on peanut allergy. So I would like to go back to that timeline of therapeutic development that I started with and illustrate what is currently in the pipeline for food allergy. So, as I've mentioned, in 2020, Palforzia got approved in the US. Viaskin, the peanut Viaskin, has been submitted for FDA approval. This is a peanut patch. There are 2 other forms of patch, including milk and egg, that are in various stages, late stages of development. We know that there are several biologics that are currently being tested for food allergy, dupilumab and omalizumab, that I went through. Shown in bright red here are different microbial therapeutics. So from the crude fecal microbial transfer to this is a probiotic plus immunotherapy. This is a defined cocktail, a sort of 2nd generation probiotic with oral immunotherapy. These are all in ongoing clinical trials. In dark red here and in black are 2 different injectable immunotherapies for peanut allergy that are also currently in clinical trials. And shown on the left are 3 different categories of other biologics and small molecule inhibitors that are already in the pipeline for other allergic diseases and could quite rapidly go into testing for food allergy. So 10 years ago, we only had oral immunotherapy being tested in small studies in academic centers. And I think now we really have a wealth of therapeutics in the pipeline. So what does the future hold? I think that as we develop and test more and more therapeutics, I think it's going to become clear that not all therapies are suitable for everyone. Some of this will be based on biology, that not all treatments are suitable for all forms of food allergy, but this will also be based on treatment goals and how the process of treatment fits the individual. Certainly, continued avoidance may be the best option for many individuals. I think on the biology front, we are making really good headway with research on biomarkers that can start to identify who is a good candidate for which therapy. This is one area where my lab has been heavily focused. We've been studying both the epicutaneous immunotherapy and oral immunotherapy trials, trying to understand what are the markers that can identify those who do and do not respond to therapy. So really, we hope to through this research, identify the best candidates for each type of therapy and for new therapies as they are developed. With that, that's my last slide. I'd like to thank the team at Food Allergy Canada for inviting me to participate in this webinar, and I would love to field some questions.
Great. Well, thank you so much, Dr. Berin. That was a great presentation, and it's very encouraging to see all the activity in the development pipeline. So there were lots of questions in advance of the webinar and that came in during the presentation. So we have some time now, and I'd like to ask you some of those questions on behalf of the audience. So the first question is around the safety of clinical trials. How safe is it to take part in clinical trials for food allergy therapies?
Sure, that's a great question. So at every institution, first of all, I want to point out every institution has an institutional review board, and the main focus of that institutional review board is to protect participants in clinical research. So each study has to really balance potential benefit and risk. This is also a really important part of the informed consent process. So the study physician will explain what is the potential benefit and what are the potential risks as part of that discussion that needs to go into whether or not this might be a good fit for you or your child. Just to mention some of the procedures that are common for food allergy studies. So right now, a food challenge is generally performed both at the start and at the end of most food allergy studies. And this is really essential for determining how much protection this therapy has produced from start to finish. There are also a number of blood samples that are normally taken. So this is both to track the response through measuring things like antibodies, but also doing research for biomarkers to try and identify, are there subsets within this patient group that do or do not benefit from treatment. I think that's going to be a very important thing going forward. And finally, I just want to mention the concern about placebo groups. So sometimes people are not thrilled about the idea of joining a clinical trial if they may get assigned to a placebo group. We know that having a comparison or a placebo group is really essential to knowing whether this therapy is better than nothing. But there have been changes made to trial design. So a lot of trials use a placebo crossover design. So individuals who are randomized to a placebo group will get crossed over to active treatment after that initial period. And of course, I want to mention that anything that is being tested, we don't know if it will have a benefit. That is why it's being tested. So certainly being on active treatment is not always the best. So really it's very important to have that placebo control group in there.
Okay, great. Well, listen, on the something that I'd like to also share with our audience when it comes to learning more about clinical trials in Canada. You can find more information on what trials are going on in food allergy therapies by going to clinicaltrials.gov, and you'll find all the listing of those trials. If they are recruiting and you fit the eligibility criteria that's outlined by the investigators, then you can reach out to them to inquire about participation. I want to move to another question that came in during the session when we were talking about the desensitization of with the Aimmune product, where the research was showing 75 to 85% of individuals had been desensitized. Now, and correct me, please, Dr. Berin, if I got that summary wrong, but the question that the individual has on this, which I think is a really good one, and it may be on the minds of a lot of people in the audience, is was there any relationship to the success of desensitization to the levels of IgE that those patients had in their blood? So, can you infer as a biomarker that if you go in with lower levels of IgE, are you more likely to have success? So if you could address that question, and I expect because of the research that you're doing on biomarkers in your lab, that might be an area that you can lend some expertise to.
Sure. That in fact is pretty much the only biomarker that we found that does seem to predict treatment success, is a low level of IgE to begin with. But there are no firm cutoffs and there's a lot of overlap. So I think we're not at the point yet where we can say, okay, this level and below you will be successful, and this level and above you will not be successful. I think that it's quite a crude marker, and there's a lot of active research going on in this area. So some of the research that's going on at Mount Sinai is looking at the IgE response in greater detail. So looking not just at how much IgE binds to peanut, for example, but where on those proteins does it bind to? And really getting a much more sort of high dimensional view of that IgE response. And it seems like that kind of mapping seems to be more predictive than just levels of IgE. So I think that there's a lot of work in that area, but certainly IgE seems to be one area where lower levels are good prognostic factor.
Okay, terrific. Thanks for that insight. So on to another question. It seems like a lot of the treatment and development pipeline is geared towards younger children. Can you speak to what may be applicable for older children or adults?
Sure. I think that with some of the immunotherapies, there has, definitely been data suggesting that young children have a better success rate. That doesn't necessarily mean that there is no success rate in older children for some of those allergen immunotherapies. I think this speaks to the fact that our immune system matures as we age and may become more resistant to treatment. Things like the new biologics that are going to be tested that, for example, wipe out IgE or inhibit IL-4. Here, that is not age dependent, that this is going to inhibit independent of age. There is no sort of response to allergen that is going to be different in young versus older individuals. For a lot of these new therapies, they are tested in teenagers and adults first as a matter of safety, and then go to younger and younger ages. So we will get an idea of what works in older kids and adults along the way.
Terrific. Okay. So this next question I will take. It's with regards to the status of OIT in Canada and Palforzia that you mentioned earlier in the webinar. First off, having an FDA-approved therapy is an important first step to advancing the conversation and accelerating the possibility of therapies for our community. Our organization has reached out to Aimmune to understand their plans for the Canadian market. At this point in time, it's not surprising because this happens with other approved therapies. The current priority is the launch in the US. That said, oral immunotherapy is currently available in Canada, but on a very limited basis. And over the past several months, the Canadian Society of Allergy and Clinical Immunology has been working on developing OIT practice guidelines for Canadian allergists. These guidelines were published on March the 18th, and they provide a broad set of recommendations that chart the optimal clinical practice and define the conditions required to bring about changes to food allergy care. So this is the first step in increasing access to this therapy. Now back to you, Dr. Berin. This question, earlier in the presentation, you spoke about a vaccine for food allergy that was then shelved as of 2013. Is there other vaccine for food allergy in the development pipeline that you see, and what's your perspective on that?
Sure. There are a number of different groups working on this vaccine type of approach. That initial vaccine was done using, and basically many of these involve the same thing. So delivering the allergens, so in this case it was the peanut allergens, they had been modified to reduce their IgE binding, although in the patients that were tested, they could still induce reactions. But they were then encapsulated by basically being produced by E. coli and were administered by the intrarectal route. So the idea that it was bacteria that were producing these peanut proteins, it was thought that it was going to be very safe, and also the route that it was thought to be very safe. The idea that it was the bacteria would provide additional immune modifying components like adjuvants that are present in a vaccine that would induce protective antibodies. So that particular approach did not work, but there are a number of basically nanospheres that are basically doing the same thing. It's a synthetic kind of encapsulated allergen. There's testing of a lot of different types of components on the surface of those to try and modify the immune response. So there are a number of groups that are still working on this type of technology. It tends to be more in academia and the private sphere, or in academia rather than by biopharma companies. But it's still active. There's also, you know, that's what I think of when we talk about vaccine, just because that is how that E. coli with peanut proteins was referred to for so long. For many of us, we also think of vaccines as being preventative. There's a lot of interest in early life interventions that can protect from the development of food allergy. So sort of three things that I can think of off the top of my head is early introduction to different foods and how best and when to introduce them. Secondly, there's a lot of interest in probiotics for prevention of food allergy. And thirdly, there's a lot of interest in the skin. We know that having eczema in early life is a key risk factor for development of food allergy. So if we go with aggressive skincare to really treat that eczema, can we prevent the development of food allergy and the whole atopic march down the road? So I think that's it on the vaccine front.
Okay, so just as you were speaking about probiotics, and this question has come up in the past, and I think we've had another question on it. Like when you hear about probiotics, you naturally think, okay, I'm going to go out and eat more yogurt that has probiotics in it. Talk to us a little bit about probiotics as in the treatment framework.
Sure. So probiotics have been around for many years. There are some of them, like bifidobacteria. There's some that have been around for many years and are commercially available. Many of these have been shown to be immunomodulatory in some form. But I think what's different about this next generation of probiotics is that they have been more rationally identified as being missing from the gut of those who do go on to develop food allergy, or through these mouse studies have been shown to actually prevent or treat food allergy when administered. So I really see this as a next generation probiotic, and likely the ones that are commercially available now, there's really little evidence that they can be directly used for treatment or prevention.
Terrific. Okay, well, listen, we could go on asking questions and more questions because there's many that have come in, but we are quickly running out of time. I do want to thank you, Dr. Berin, for this really informative session and for answering our questions. I also do want to let people know that have more questions as it relates to oral immunotherapy in Canada and how to think through that treatment is we have a very good webinar on our website featuring Dr. Julia Upton. You can find that webinar in the tools and download section under research. And I would encourage you to take a look at that because it really is a good, careful look at who it might be right for and who it maybe isn't best for. So thanks to everyone for your participation and submitting your questions. Dr. Berin has really helped us understand that the food allergy research landscape is looking very good. There's lots to be optimistic about. And to that end, one of the roles at Food Allergy Canada is to make sure that you get the latest information on food allergy, including progress in research, and we'll continue to follow the developments and treatments and therapies and keep you informed. Also, please stay tuned for more details on creating a better future webinar series. There will be more, and there will be another one that digs deeper into immunotherapy in this series. And lastly, you're going to get a short survey through the GoToWebinar platform, and we would ask you to take some time to fill out this survey, It really is helpful in making us understand what's working, what's not working, and inform and improve our webinars in the future. And we'd also like you to stay tuned for details on creating a better future. And we'd like to thank our sponsors for the webinar series, Pfizer Canada, Sean Delaney Memorial Golf Classic, the Walter and Maria Schroeder Foundation, and the Peanut Bureau of Canada. Thank you for participating in today's session, and we wish you well in the coming days and weeks. Thank you.